The first part of this lesson up here with these two hemispheres is actually going to be kind of fun. I think this is what people really want to know about the brain. They want to know things like where is your sense of time, or your math skills, or smell, or speech, or what controls your muscles, things like that. So we're going to divide these up into lobes and add a little bit of color and just write what goes on in those lobes. So first you'll remember that the cerebrum is divided into two hemispheres. From the top, the two hemispheres look like this in our very first drawing with that longitudinal fissure going down. Now, from the inside, if we take a slice right across the middle in a cross section, that would be cutting here, and then looking at the end, it would look something like this. So you imagine you're looking straight at somebody, the eyes are here, the nose is here, and you've cut the brain, and you can see this is a hemisphere here, and this here. So this is like sort of empty here. So this would be the corpus callosum connecting. You see there's just this little bit connecting them, and then down here they look a bit connected. But here, this is separate and up into here. So the two sides of the brain are only connected at relatively small places. And these right here, this space here is a ventricle. We'll get to that in the bottom part of the drawing. And the exterior, which is really what we're going to see in these pictures, looks like this, more like that in color. And you can see this lateral sulcus here and here. Everyone's brain has this, so that's why I've included it here. That's always visible. The other ones, like the central sulcus, I don't know, is that one? Looks like maybe that's it. Here, could be this, or it could be this. They aren't so clear. This is the only sulcus that's really clear on everyone's brains, and that's the one that you have right here. So we're not going to worry about any other details except for imaginary ones that we're going to draw. So we're going to draw lines that aren't part of the actual brain. This one is, but everything else is just going to be imaginary lines. And our lines will look something like this. We're going to add a little bit of color, except I don't think we'll color them in. I think we'll just kind of outline them in some colors. So these will tell us areas of the brain where certain things happen. All right, so that's kind of where we're headed. We're going to do, obviously, the left side. You could imagine if you want to draw a little eye in, you can see how a person is facing like that. The ear would be here, so you're looking at the left side of their head, and this is the right side. So let's zoom in on the left here. And they're going to be very similar, although we'll see some differences. Okay, first thing to do is draw a light line right like that. And then draw a line here and here. Then go along here and we're just going to kind of round it out like this. And then draw a line kind of coming back here like that. And then if you'd like to, you can do the same thing over here right away. Draw up and down. Two thin lines like this. Round that out to make the temporal lobe. And then go like that. And they don't have to be perfect. Okay, I'm going to go back over to the left one here. And I think I'm going to use a little bit of color to outline them. And I'm going to make my colors match the hat, which is one of the activity options that goes with this lesson. You can download, cut out, and assemble a brain hat. So I'm going to make my colors match that brain hat. I'm going to take an orange and just go around the outside of this. If I completely color it in, then it's going to be hard to do any labeling, writing any words. And the next one I'm going to outline in green. And the colors really don't matter, though. You could really use any color because obviously there are no colors in the brain. So make my next one blue. And this nice big one I'm going to make yellow. And actually, this blue one is sort of it's considered part of this lobe. This is the parietal lobe. So the blue one is kind of like a subsection. But I think it's better to go ahead and learn them as separate parts. But we'll mention it again that the function of this blue strip is very much related to the whole lobe. And then I think I might use a purple for this back one, which is going to be our lobe that controls the eyes. Okay, and then I'm going to use red to outline this one. And then this other stuff down here, if you want to add some stripes, this is the cerebellum, right? If you want to add some texture to it, you can. This is the pons, cerebellum. Now, what are their names and what do they do? Let's do the red one first. This is the temporal lobe, and you can see I'm going to draw some little, like a nose and an ear and mouth and some things like that. You don't have to. You can just write words if you want to. Let's label this as temporal, just like it sounds, temporal lobe. And it's named temporal after the bone that it sits behind. This is the temporal bone. So the temporal lobe is found right behind here. And as long as we're looking at this, let's look at this too. See the parietal bone. Guess what this lobe is going to be named? Yep, there's the occipital bone for the occipital lobe and the frontal bone for the frontal lobe. So they coordinate with the names of the bones. So we're kind of learning names of bones along at the same time. Okay, so the temporal lobe has smell. I'll put a little nose here, put smell. Now you remember we saw those olfactory bulbs coming in on the bottom. If we look at the bottom of the brain, we saw those olfactory bulbs coming in there. So they're going to come in and attach to a place in this lobe here. And also let's put an ear. Now, which ear am I going to draw? The left temporal lobe is connected to the right ear. So if we can imagine on looking at the right side of the head, the ear is going to go this way, actually, like that. Now I'm going to right ear. Okay, and in the left temporal lobe, we also have speech. I'm going to make a little pair of lips there. Now this is one of the things that's going to be different about left and right. Most people have their speech centers on the left side, maybe about 90%. 10% have it on the right. So that's another thing the brain surgeons have to be very careful about. You've got to make sure that they know if they're going to be operating any of this area, that they know where the person's speech center is. Now, this is also connected to an area right here. This frontal lobe. There's a little area right there called Broca's area, named after Paul Broca, who was a brain doctor. B-R-O-C-A. And then if you want apostrophe S, it's Broca's area, as if it's belonging to Broca. Broca's area, and let's write it down here. I'm just going to erase my little eye here. Let's make a note about it right here. B-R-O-C-A. Broca's area is where we do forming sentences. The fact that you can put words together in an orderly way by the rules of grammar and form a sentence, that's where it's happening right here. Obviously, if something goes wrong, if you have a tumor or you have a stroke here, that's what's going to be affected. You won't be able to put words together. You will still be able to understand sentences, you just won't be able to make your own. Understanding sentences happens here in Wernicke's area. And I'm going to write here first. W-E-R-N-I-C-K-E. Wernicke. Wernicke's area is where we have understanding sentences. So if you can write really small in there, Wernicke's area. So obviously these two work together a lot, because as we're forming sentences, we're then listening to somebody else speak and understanding. This was actually one of the first brain parts figured out. I think this goes back maybe over 100 years. Paul Broca lived quite a long time ago, and he had a patient that couldn't form sentences properly. And then after she died, he looked at the brain, and there was a very big wound here, a lesion right there in that area. And he noted that. He made a little diagram and said, right here, that's probably the area of the brain that controls being able to form sentences. So that was one of the first ones we had figured out. Okay, there's a few other things that the temporal lobe does. It is somewhat involved in emotion and memory, and people that have seizures in the temporal lobe sometimes have very, very emotional experiences, and sometimes even religious type experiences. That's the way they feel. So it's not exclusively just speech, hearing, and smell. There are some other things that go on there, but that's the important ones to know: speech and smell and hearing. So let's do the frontal lobe next. We just met one area of the frontal lobe. Let's go up here and write frontal lobe. This is where all your classic thinking goes on. Your math skills, logic, decision making, things that you really think are thinking. Your conscious thinking. So let's write, I'm going to start down here and just go up. Math, computation, logic, analysis, comparing things, words, what they mean, time. This is where your timekeeping is. Understanding time and how much time has gone by, reading clocks, all that stuff. Also lots of decisions are here. And additionally, the frontal lobe is responsible for some of our personality, especially our moral decision making, the fact that we can understand right and wrong, and things like compassion and responsibility, our ability to show up for work on time, to not cheat on tests. A lot of that kind of behavior is centered in this lobe, and we know that because we saw someone that had an injury, a very famous person that had an injury to this part of the brain. And his name was Phineas Gage. He lived in the mid-1800s, and here he's holding a big metal spike, a tamping iron. he worked on the railroad, and he would use this to tap explosives down into a hole. They were blasting to remove dirt and rocks to put in the railroad spikes. And so he would put explosive down there, tamp it down, and then they would clear away and light it. But unfortunately, you can hear this one coming, can't you? Yeah, so he was tamping with his iron, and it exploded, and unfortunately, he was standing over the end of it working, and the spike did this. It went suddenly up, pierced right here, went through. I mean, the drawing says it all, doesn't it? And you're thinking, how could anybody survive that? Yeah, that's quite a question. How he survived it, it's just amazing, really amazing. He was sick for quite a while. You could imagine with gaping holes like this. Back in the days before they had antibiotics, he had to fight off quite a few infections. So he was pretty sick for a couple of months. But then after about a year, he seemed like he was fine, except for this eye is affected. Obviously, it had a pipe go through it, so he couldn't see out of this eye. It closed. But otherwise, he seemed pretty normal. He wanted his job back, but they said, maybe not. So he ended up doing some other things. The most famous of his jobs was he went to Chile, like the country in South America, and drove a stagecoach for a while. Kind of crazy. But as time went on, they found out he wasn't quite as fine as he thought he was at first. And the major changes were that he became a not-so-nice person. He became kind of nasty. He started swearing, using bad language, drinking, making bad comments about people, not showing up for work. And before the accident, he was just your most Mr. Wonderful. He was responsible and nice. Everybody liked him, friendly. He was just your super nice guy. And after the accident, he was not the same person. And the famous quote by the doctor who studied him said, "Alas, Mr. Gage is no longer Gage." He just wasn't the same person he used to be. So we know because we have the skull, and just don't ask how they have the skull. Yes, they had to go dig it up. But they have the skull and the tamping iron still in a museum. It's in, I believe, in Massachusetts, in Boston, in an anatomical museum. And so they can study this, and they've got all kinds of brain images. You know, they've made 3D animations of it because it's just fascinating. And so they know exactly where the pike pierced, and it went right up through here. And there's some videos about Phineas Gage. If you want to learn more, you can go to the playlist and see a little bit more about him. The next part, the part I've done in green, is kind of part of the frontal lobe, but we're going to label it on its own. And it's not called a lobe, it's called a cortex. It's called the motor cortex. I'll see if I can get in just a little bit closer here. Motor, M-O-T-O-R, cortex. Now, motor means movement. So this is the part that's connected to the muscles. So when you decide, I would like to move my arm, this part decides, this is where your decisions are. Move the arm, it relays the information to here. And then this part, of course, we've seen already, it feeds down through the midbrain, and it has some connections here, and you have to have dopamine, and then it goes down, out through here, and then out to the muscles. The next little strip is called the sensory cortex. And this is sort of connected more to this. And this is where you have the incoming signals from the senses. So you remember we have coming in through here, through the spine, all the nerves collecting information from the hands and the feet and the toes and the back and everywhere else. And then it comes up through here. It gets sorted out. As we saw, you get the thalamus and the midbrain and everything. And then the signals that make it all the way up come into the sensory cortex, and then they can be relayed to the bigger part of the lobe. So this is where they all come in, and then they go to the parietal, P-A-R-I-E-T-A-L. The parietal lobe, and that's because it's under the parietal bone. And this is the lobe that keeps track of 3D space, where your body is in space. You know, you can close your eyes and move your hands around, and you know where they are, right? You don't have to see your hands and feet to know where they are. I could say cross your legs, or bring your hands together, touch your fingers together, and even with your eyes closed you can do that, because your parietal lobe is keeping track of all this input coming in about where the arms and legs are, is keeping track of it and making sense of it. So I kind of sometimes like to make a 3D cube. You know how to make that, make a square, make some lines going back like that. So this keeps track of 3D space and where the body is in space. Body, could say body awareness. And beyond that, it also understands 3D space in a kind of abstract way, like in geometry with mathematics. This is where a lot of your understanding of geometry is. You understand what a cone is. You understand what a cube is. You understand what icosahedron is, right? Icosahedron? Okay, but all these shapes, you understand how they can be 3D. You can turn this around in your mind. Let's say I put a dot on one side of the cube, and then I say, now, in your mind, spin that cube so that you can't see the dot anymore. And in your mind, you could turn that cube around, push it so the dot's facing the other way. This is the lobe that lets you do that kind of thinking. A little trivia about the parietal lobe is this is the only lobe that they think that Einstein had an advantage in. His brain just looked like everyone else's brain. It didn't have any more neurons than anybody else's. But this area of his brain, there's a little bigger parietal lobe. More of his brain was devoted to understanding 3 dimensions. And if you connect that with math skills, you're able to imagine math sort of like 3-dimensionally. I know that kind of sounds strange, but it was this area of his brain actually that gave him the edge, not the little logical calculations. Now I want to go back and say something about these cortex areas. So if we were to slice right here and then look at it this way, we would see something like this. We looked at this before. And then if we drew just the gray matter like this, here and here, we would get a map of those cortexes, and it might look something like this. Now this is a type of diagram you'll see very commonly in any brain curriculum, and they've helpfully color-coded it down here so that the red is showing you the motor cortex and the blue behind it, that's showing you the sensory. But they're very much the same. And there's always this little funny looking body part things lined up along the top of it. You see a foot, knee, and hand with no head. The head's down here. Huge, huge head. There's the tongue. And over here on the sensory, we even have teeth and gums. We have these gigantic facial parts, and then these little tiny little body parts like that. So what's going on here? Well, this is showing you what part of the cortex is attached to which body part. And so it becomes very obvious that a very large section of the cortex is devoted to the eyes, the nose, the lips, the tongue, the mouth, and the hands also. Hands are very large. And this makes sense because we interact with our world very much through our eyes, through our speech, and through our hands. And so we need a lot of feedback. You know, your fingertips have to be very sensitive. Your knee doesn't really need to be that sensitive. You need many more neurons attached to your fingertips than any of these other body parts. So anyways, the cortex has an appropriate amount of neurons dedicated to the parts that need them. Now, I'm going to show you a picture of how artists sometimes take this idea of the proportions being unusual, large head, large hands, and they actually draw a little person with the correct proportions. And this are both called the homunculus. You can call this the homunculus And you can call this the homunculus, which is Latin for little man. Now, this is literally a little man, whereas this is kind of body parts strewn along the top of a cortex. But I thought I'd just mention that it's called the homunculus. In case you see that word in a brain curriculum, you'll know what it means. Okay, let's finish this last lobe that I've made purple. This is the occipital, O-C-C-I-P-I-T-A-L. So you have a hard C, k, followed by a soft C, s, occipital, occipital lobe. And this is connected to the eyes. Now, this is the left hemisphere, so it controls the right eye. So I'll just make a little eye there and label it right eye. Now remember, we looked at the bottom of the brain and we saw where those things cross. So coming input here and it crosses over like that, and the input from this side crosses over and goes like that. That's the optic chiasm, and that's where the lines cross and go to the opposite sides of the brain. Now let's go up here and sort of summarize the things that the left hemisphere is known for. First of all would be speech. And you could put 90% of the time, or 9 out of 10 people. I'd say the second thing it's most famous for is math calculation. Now, not understanding geometry, but adding, subtracting, dividing, all that good stuff. That's where your arithmetic is. And then kind of related to that, we have logic, and I'm going to put and analysis. And that lets us compare, figure things out. This is your troubleshooting skills. Another important thing is sequencing, figuring out cause and effect, what comes after what. You can see a series of pictures and put them in the right order to figure out which happened first, second, third, things like that. So sequencing and seeing things in parts. The left brain is very good at looking at the individual parts of something and figuring out how they go together. It's also where we have our time, sense of time, both understanding time and feeling time. Like if I say, I'm going to start my watch, tell me when 1 minute has gone by, and you have to guess how long a minute is. That would be a left frontal lobe function. And let's also put in symbols. The left side of the brain loves symbols. Like I can put a little stick figure, just a circle with a few sticks, and you know exactly what that's supposed to be. Or like a heart stands for love, and you know, a star stands for something good, a smiley face. I mean, all that is a circle, 2 dots and a line, and you understand that it's a face. It doesn't have to have hair or ears or anything. So the left side of the brain loves to let symbols stand for ideas or words. So let's move to the right hemisphere, and we're going to have those same areas. Let's use the same colors. And I'm going to label the pons again here. And we've got the cerebellum. Don't worry about which way the stripes go. Which I think's good. So by now we should really getting familiar with these basic parts like the cerebellum and the pons, and of course this is the brain stem down here, medulla oblongata. Okay, again, let's start with the temporal lobe. And we've got smell here again. You can draw the nose. You don't have to draw left and right nostrils, just nose for smell. And we also have hearing. So we would be drawing this ear. Now we don't have the speech. Let's pretend we're a normal 9 out of 10 person. Don't have speech here. But you do have a little bit of a contribution to speech. Right? I don't know if it's right here, but somewhere around it here. The right temporal lobe adds rhythm. We're going to find here in the frontal lobe that the right side of the brain is all about music, art, creativity. So what it adds to speech is the kind of musical quality to it. You know, you can make a sentence go up at the end to sound like a question, and there's a rhythm to it. This would be where you would be able to recite poetry. Humpty Dumpty sat on the wall. Humpty Dumpty, like that. This is where you have rhythm of speech. Now, as strange as it might sound, if you have an injury or a stroke or something here in the rhythm center, you would be able to form sentences, you would be able to speak, you would be able to understand sentences, but your rhythm would be a little bit off. And then I'm going to add here also it plays a part, it does play some parts in memory and emotions, just so we know that the temporal lobe isn't exclusively about hearing, smell, and speech. There are some other functions there. And let's go up and label frontal lobe. And here we're going to have, well, I've already mentioned, it's best known for creativity and music and all things art, sculpture, drawing. This is where your drawing skills are. Even if you don't have that many drawing skills, you still have some, and that would be where they would be. And also the right side is responsible for intuition, and I would just say insight. And then also I'm going to put here, I'm just going to write the word eureka. This is where you have a flash of insight, and all of a sudden you're like, oh, I get it now, I get it. You just have a great idea, or you just suddenly understand. So, and if you have space here, we can also put seeing the whole picture. Seeing the whole. The right frontal lobe sees the grand scheme of it all. It's like you have an eagle-eye view on whatever you're doing, a project, your future, whatever it is. It sees the whole picture. Whereas the left side sees little individual parts. So a lot of times you have to make them work together. When you're doing a project, you need to envision the whole project. You need to see where you're going. You need to understand the end result. And then you need the left brain to kick in and say, okay, now where does this gear fit in? How am I going to stack these little pieces together? So life is kind of a combination between left and right. It's the way it's supposed to be. All right, so here we have the same, exactly the same as the other side, motor cortex and sensory cortex. So we could also add a note here, though, since this is the right one, we could put, make sure we remember left side, and then over here we can make a note. right side. And we don't need to discuss this further because we already did. Here we have parietal lobe again. And if you want to draw your little nice little cube again, your body, we've already discussed that. So this does the same as the other side basically. There might be a few differences, but nothing so major that you can find lists on the internet or anything. Basically same thing. And we have the occipital lobe back here, and of course it's going to control the left eye. Remember, this is the part that turns the picture right side up. When the information comes in from the eye, you see an upside down image, and this is the part that turns it around and actually sees. Alright, now let's do this list here. What is the right hemisphere known for? And you can find lists like this if you search the internet. There's lots of them. The number one thing it's known for is creativity. That'll be on everybody's list. And let's put art. That'll be on everybody's list. And music will be on everybody's list as well. And let's put flashes of insight. Flashes of insight. And if you want to write Eureka, E-U-R, that's what they say. The Greeks said, I get it now. And what also the whole, seeing the whole picture. I don't know how better to say that. Seeing the whole picture. And we also said rhythm of speech. Rhythm in general, because that will be part of music, but also it contributes rhythm to your speech. Okay, let's come down here and talk very briefly about one type of memory. The topic of memories, how they're stored and how they're created, is very complicated and not entirely understood yet. There's a lot of research going on. We know a little bit, but fundamentally, any neuroscientist who's honest will say memory is still very much a mystery. So we're just going to look at a few things that have been discovered about declarative or explicit memories. So you can see right up here I've got the word declarative / explicit. Now these two words can be used interchangeably. They mean the same thing. Declarative is the type of memory that we think of as a memory. When you remember your math facts, when you remember the vacation you had at the beach last year, when you remember what your friend said to you 2 days ago, that kind of memory. We're not talking about muscle memory, remembering how to play the scales on a piano or remembering how to ride your bike. We're talking about a declarative or explicit memory. Now, the opposite of declarative is non-declarative, and the opposite of explicit is implicit. And then under this category, you can subdivide into other types of memory. You can say there's episodic, there's semantic, there's all kinds of words you can learn for classifying types of memory. But we're not going to worry about all the different definitions. This is just a really basic overview. So we'll get to this in a minute about what short-term and long-term involve. We'll get to that in a minute. First, let's give this head an eye. Draw a line like that. Roll around dot. Kind of an oval. Like that. And then give it some kind of a nose. And then a mouth. Just go out, in, out, in, and then kind of around for the chin. And don't spend a lot of time drawing this. This is it's not important to get this looking really right. If it even just looks remotely like a face, that's fine. So we need the face because we're going to start with sensory input. We also have a little piece of spinal column coming up. That's going to give us some sensory input. We have an ear. That's going to be some sensory input. So most memories start out as sensory input. Let's draw over here from eyes, nose, from mouth, ears, up, from the body. Now some of these we could draw an arrow like this, would go actually straight to the cerebellum and not get processed by the cerebrum. But we're going to talk about the ones that do go up here, and we'll put a number 1 here. So all these inputs, they're electrical signals, they're action potentials coming in from the neurons in the body, and as we already mentioned, they first go to the central area of the brain, the thalamus, and it's a sorting center where they get sorted out. And some of them aren't going to make it through. Remember, your thalamus has to decide which ones are most important. And it can only retain about 12 sensory impressions at a time. Now, that's just pretty amazing that it can retain 12. But this would include, like, for example, right where you're sitting right now. You're listening to my voice, that would be one thing. And you're understanding what I'm saying, that would be another. You are looking at your paper or you're watching a video, so what you're seeing is another one. And we can even subdivide what you're seeing. You're seeing colors, you're seeing shapes, and you're seeing things out of the corner of your eye. You're probably aware of other things in the room. You may be hearing different sounds. Maybe there's a dog barking outside, or you're aware of how your clothes feel in your body. Maybe a cuff of your shirt is squeezing you too tight and you're uncomfortable. So we have all these sensations coming in, and we have also then our own thoughts about the sensations, but we're keeping this real simple. The senses come in, and so let's put above the 1, let's write sensory. So your brain holds very temporarily about 12 sensations, and then it's going to pass some of them along. Now, you might be saying, wait a second, I thought that the occipital lobe processes the information from the eye. This is true, so let's make a little over and back arrow. Sometimes this information has to make a quick trip somewhere else. I'll write the word I there, because your thalamus would probably like to have the image right side up, so it can decide what to do with the right side up image. So real quickly, it can go over, it can get flipped around, made sense of, can come back to here. Same thing with some of the things from the body. Maybe they'll make a quick trip up to the parietal lobe and get interpreted and come back to here. And in the end, all the brain parts are connected somehow. We have this just lightning back and forth going all over the place. So we're ready to send some of these sensory impressions onto short-term memory. And over here, we could write a number 2, and then the words short-term. And this really is in this area of the brain for this type of memory. Sometimes they call it the prefrontal cortex. If you want to write prefrontal cortex. So remember, the cortex is this, and it's the prefront. That kind of doesn't make sense. How can it be in front of the front? But at any rate, it's this very frontal part. And so this is like our clipboard, where we can store up to 7 things for a few minutes, maybe a few hours, but think of it as just a few minutes. And the amazing thing is that no matter how smart you are, you still can only store 7 things, maybe 8, like 8's the world record, but this little clipboard, short-term memory, only can store about 7 things. So if you want to keep them longer than just a very short time, you're going to have to transfer them to long-term. Let's make number 3. Long-term. And long-term, don't think of like years. Some memories do last years, and that is long-term. But long-term is also trying to remember the answers you're going to need for a test tomorrow or next week. Long-term storage begins with hours and days. So let's see, let's make our arrow go up to here. Goes to the short term, and then over to long term. So then it goes over to long term, and the way that that gets recorded in long term is by these parts in the middle let's draw, it's going to be the mammillary body. We'll kind of curve this around, draw these limbic system here. Will be the hippocampus. There's your amygdala. So I'm going to write A for amygdala, H for hippocampus, F for fornix, M for mammillary body. And these all play a part in memory. Amygdala is concerned with emotion, but remember, emotion often strengthens memories quite a bit. So we get like a memory boost here from the amygdala. And remember, the mammillary body, it's where you remember spaces, how to navigate and where you've been, things like that. So then the hippocampus is going to move these impressions out like this to the cortex, or neocortex if you prefer, and it's going to break things up into pieces, unless you're remembering one particular item. But let's say you're remembering your birthday party last year. Each part of that experience would be stored separately. The things that you saw would be stored probably in the back here near your occipital lobe, which deals with vision. The things you heard, I mean, maybe part of it would be in the temporal lobe. And actually, I must say the temporal lobe here, which isn't shown and we're not talking about, it does play a significant part in a lot of types of memories, but we're just keeping it simple here. And so some other aspect of the party would go here, how you felt about it, what people said to you, the presents you got, whatever. Each part would be stored separately, so that when you want to remember that event, the hippocampus is having to pull those out, kind of reverse the process. We could make it a dashed line going over here. And it has to gather those parts, reassemble them, put them back into your short-term area so that you can look at them and think about them. So this is your conscious, what you're thinking right now. Just to put them back into there. Then when you're done, it goes like this and it puts them back again. So like we mentioned before, you can kind of think of the hippocampus as the librarian that puts things into storage and gets them back out. So let's talk about what this means. Short term involves glutamate crossing synapses. Long term involves production of new spines on dendrites. All right, what is all of this? Well, we have to remember what neurons are. They have the cell body with the dendrites, an axon, and then they go into axon terminals. These little bumps here are the terminals, and then they connect to another neuron. There's the dendrites again that goes off this way, and here's the previous neuron coming in like this with its terminals. So we only see one complete neuron here. And between the axon terminal and the dendrite, there's a little tiny gap called the synapse, like this. This would be the axon terminal, and this would be the dendrites that are going to receive the information. And you remember that what crosses the gap are these little chemicals called neurotransmitters. And in this case, involving short-term memory, the neurotransmitter is glutamate, which is a form of the amino acid, glutamic acid. And over here, I just wrote a little diagram. Glutamic acid has COOH. It has the carboxyl banana hanging off. Actually, there's one on either side. And so it's got this hydrogen. When the hydrogen leaves, then it looks like this, and it becomes glutamate. So you basically have the amino acid glutamate in here acting as a neurotransmitter, and I'll bring it closer so you can see it crosses the gap and it sticks to some receptor sites on this ion channel. And this ion channel will let sodium and calcium ions go through. When it's activated, it will let these sodium ions rush through, and that's what's going to start the action potential in the neuron. It's going to restart the electrical signal. Now there's one of these types of receptors that has a little magnesium atom clogging it up. Magnesium sticks here and prevents things from going in, but with enough influx, very, very strong influx of glutamate, you can dislodge that magnesium, and then calcium will rush in, and then calcium will start some other processes too deep inside the cell that will initiate stronger memory. So this process is pretty temporary, all these little things crossing the gap. This is the only mechanism that's involved in short-term memory. And to get memories to really stick a long time, you need to change the dendrites. And here is a picture drawn by Ramon y Cajal, if you remember him from the nervous tissue lesson. He drew this picture of a dendrite, and you notice it looks furry. So here's some new information about dendrites, and you'll find this is almost always the case in science. No matter how much you know, there's always more you can learn. And here we learn that dendrites actually have little tiny spines sticking off like this. And he saw them way back in the 1800s. He didn't know what they were yet, but he knew that some had more than others. So here's an actual picture, an electron micrograph showing the little spines. And down here is a little picture that shows the growth of a spine. For that, let's go up and use a Wikipedia picture. This series of pictures shows the growth of a new spine. So here is an axon. This is a dendrite. The dendrite already has a couple little spines here, but it's going to grow a new one right here. There will be some kind of signaling going on here, and the dendrite will know, hey, I better go find out what's happening down here in this axon. And it'll put out a little thin spine. This gets a little bit larger when it makes contact, and then it's going to get a little bit larger, and this is going to get a little bit larger. And if it doesn't get used, it's going to start to shrink. But let's say this one gets larger and larger, and eventually it's going to become very well established and look like this. So here's a little starter one. It grows out, and then it makes contact, and it's going to start to grow. This is cytoskeleton growing inside, and the cytoskeleton is going to push and make this thing expand. You're going to have, membrane being added here. And then eventually you're going to have brought into here some little manufactured glutamate receptors. They're going to come and stick into the cell here. And these are little adhesion molecules, little places that can help it stick on to the axon. So now we have a fully developed spine, and its job is to provide extra glutamate receptors. Now, because this is going to stick like this, it's going to become a permanent sticking place, then now we have a whole new pathway opened up. And a memory is basically a unique pathway from neuron to neuron, from dendrite to axon to dendrite. There's a unique pathway that a signal can take, and it's meaningful. That pathway is unique to that particular meaning in the brain. And so, of course, you have billions and billions of these pathways. And the more the pathways are used, the stronger these connections become. So you want a lot of little spines making good connections. So when you're practicing those scales on the piano, your neurons are actually growing new little spines. In fact, while you're listening to this right now, your neurons are busy, hopefully growing new spines so that this information can stick for a very long time. Now let's go over and talk about something very different, and that is the ventricles, the empty places in the brain, which actually are extremely important. They're not just empty places. So let's look at a simple diagram that will show us where they are. Maybe I can get both of them on here. This is a side view and a front view, because they're very strange shapes, very difficult to understand in 3D. And actually, I think I've posted a video on the playlist that shows the ventricles. It turns them around three-dimensionally so you can really understand the shape. So here we see from the side, we have a couple of big ones that look like this. They go around like a C with a little hook on the back, and these things are called horns. Actually, all this end, this end, this end, they're all called horns. And then we have one right behind it, which you can't see. This horn sticking out is from the one behind it. So this horn belongs to the front one, and that horn belongs to the back one. And we can see right here, that's part of the back one. And if we look to the side view, you can see that they're actually very far apart. It doesn't look that way in this diagram, but you can see the actual shape. These are the horns sticking out. And then in the middle, they go down like this and connect to this third ventricle right here. And you see that little pink dot right there? That is where that thalamus connecting tissue goes. So the thalamus sits on either side of this ventricle, and that little connecting piece goes right through the middle like that. And then the third ventricle connects down here to the fourth ventricle, and we labeled that in the last drawing. We saw the fourth ventricle down there towards the brainstem. And then this has a couple little outlets that connect. We'll see in a minute why those are there. And let's just look at another picture while we're at it. This shows that the ventricles are all connected. They've used blue for fluid. And also, though, that they are connected to a thin space that goes all the way around the brain. See how the light blue goes all the way around the cerebrum? And it connects into here. These things go out like this, connect, and the down here goes around the cerebellum as well. And this dark thing here is going to be a vein. This represents a vein that's going to absorb the extra fluid. And these little things right here, these are little places where the arachnoid layer bulges out a little bit, and that's going to be the drain points. Now this is very, very out of scale. These veins are very tiny, and this space here, it looks big, but it's extremely small. You wouldn't even notice it. You know, we're looking at those photographs of brains. You can't see this at all. This all happens in those meninges. The dura mater, the arachnoid layer, the pia mater, all this happens in that little thin covering. But we have to make it bigger or we won't be able to understand the drawing. So we make them huge and out of scale so we can actually see them. So we'll be drawing that in just a minute. And there's a flow to it. This drawing shows you some arrows. We'll talk about that, how the direction of the flow of the fluid in here. It goes from number 1 and number 2 down through 3 and 4. And when it gets to 4, it can either drain down or it can take a ride around the brain and then drain out. So we'll put that in our drawing as well. Now, one more picture before we start. Where does the fluid come from? We've actually met the cells already that make the fluid. Remember this drawing? Eons ago? These are the ependymal cells that line the ventricles. So this would be the fluid. We're on the edge of the ventricle here. And they have little hairs, little cilia sticking up. They make fluid, and they can also push the fluid along. And that's what helps the fluid move down from 1 to 2 to 3 to 4, down and out. So it has these little brushes that can sweep the fluid along. And these are epithelial cells. They're not neurons. So the body's a mixture of tissues. You need some epithelial cells here mixed in with the neurons. There's a good review. Want to look at those? Remember the neuron. Remember the astrocytes and the microglia and the oligodendrocytes that put the myelin sheaths. Remember, there's no Schwann cells in the brain. There's these oligodendrocytes that cover the axons. And we've got the pericytes here surrounding the vessels. So it'd be good to go back and review that while you're studying the brain. Before we finish the picture, let's talk about some of the functions of the ventricles and the CSF. Ventricles are filled with cerebrospinal fluid, or CSF. And this is a very common abbreviation. Often you'll just see CSF in a text and you're expected to know that it's cerebrospinal fluid. And this fluid is basically like blood plasma. You can just put a note here. Similar to blood plasma. You remember that blood is mostly water. Over 90% of blood is just water. So that's what CSF is most similar to. It has a few extra nutrients, but think of it as blood plasma. So all the ventricles are filled with this CSF, plus you find it around the outside of the brain, around the outside of the spinal cord, going down the middle of the spinal cord, some places like that. Ventricles and CSF help the brain by providing, number one, buoyancy. Here's a hard word to spell. B-U-O-Y-A-N-C-Y. Buoyancy means floating. So you can put in parentheses, the brain sort of floats. Hard to imagine your brain floating inside of your skull, but it's true that this fluid helps take the weight of the brain off the parts below, especially the blood vessels down below. So it helps the brain not press so hard down on the things below it. Also, cushioning. And this is basically protection from bumps. Whether you play sports or not, there's times in life when you hit your head. So the CSF helps provide a little bit of cushioning. Now, of course, this is really in the news recently with all the research they've been doing on concussions and the long-lasting effects of concussions. They're a lot more serious than people had thought. So sometimes the cushioning just isn't enough. You bang your head hard enough and you're going to get a bruise on the brain and give you a concussion. The third thing that it does is to provide nutrition. So there are sugars and oxygen in the fluid. And I know the blood vessels are the primary delivery system for nutrition to cells, but the CSF does a little supplemental nutrition. Food for cells. So any cells that come into contact with this fluid can extract things that are in it. If there happen to be sugars floating by, they can take it out. And fourth, we have communication. Now, this might seem strange. How would you have any communication going on in watery fluid? Well, the watery fluid is providing a pathway for hormone messengers. Hormone or other chemical messengers. So the primary route for delivery of chemical messengers is the bloodstream, but this is another way they can get around. If they need to go somewhere else in the brain, they can float around, so brain cells can kind of talk to each other a little bit. I don't mean literally talk to each other, but they could signal if there's some foreign invader, or if one of the brain parts is injured, it could signal other cells. You know, cells do communicate. So things can float around in this fluid and aid in communication. Now let's go down and fill in the color. I think we're about ready. And I'm going to use a light blue and a dark blue, and then I'm also going to use a red. I'm going to start with a light blue and sort of obviously use this to color the ventricles. Just as an interesting note, the way that the ventricles were first discovered, I don't know what year this was, it's been decades ago, maybe longer than that. Somebody was tearing apart a cow brain, and they noticed that there was kind of spaces in the cow brain, and they wondered what those spaces looked like. So they had the great idea turning the brain upside down and pouring hot wax into the brain, letting the wax cool so it's going to harden in these spaces, and then removing the brain. And in that way he made a model of the shape of the ventricles. I don't know that was ever done with a human, but anyways, that's how ventricles were first discovered. Alright, so just color kind of around the outside so we can do some writing on the inside. And let's label these. Let's make this number 1 and number 2, although they're rarely labeled as 1 and 2. They usually are just labeled as the lateral, L-A-T-E-R-A-L. Lateral means side, the lateral ventricles. And you have to know they're 1 and 2, because when you get to 3 and 4, you might go, wait, where was 1 and 2? And they were labeled as lateral. So as we saw, that they go quite a bit out to the side. Then there's just third in the middle. There's just one of them there in the middle. And remember, that little hole is where the two sides of the thalamus connect. And down here in this area, this is where the hypothalamus and pituitary sit, down here. And this, back in here, this would be where those parts, remember, where your visual reflexes are. That's where that is. And then let's label this fourth, down there. And they don't have any other name. People just call them third and fourth. All right, so while we are taking a look at these, before we go on and do the outside of the brain, take a red or an orange or something, and we're going to color code. See down here? I'm going to mark a square like that. And we're going to color the choroid plexus. Choroid means colored, plexus means complex or network. These are epithelial cells that produce the CSF. And these are the cells that we already met in this drawing, right here, with the little cilia hairs so that they can push the fluid along, make it move. And all together they're called the choroid plexus. The individual cells are called ependymal cells. And they are located kind of on the bottom of 1 and 2 like this, and on the roof of the third, lining the inside, and a little bit here in the fourth. So most of your production is going to be in 1 and 2. And the flow of fluid basically goes from 1 and 2 down into 3, down into 4, and then out. However, it's a little more complex than that. You see these two little things here? Well, the fluid, when it comes down, can go down and out, or it can take a detour. There's two little things coming off of here like this. This won't be very precise, it's kind of approximate. Come over here, and then they join into the space that goes around the cerebrum, and this would be what they call the subarachnoid space. Remember in the last drawing we drew the meninges, the dura mater, the arachnoid layer, and the pia mater. Well, this would be right under that arachnoid layer. It's called the subarachnoid space. Now, the line that we're drawing here is way too large. This is ridiculously large. The subarachnoid space is like microscopic, basically. So this is way, way too big. However, if we drew it microscopic, we would not be able to see it. So it can flow around here like that. It also flows around the cerebellum. And it'll flow around the spinal cord, down and around. In fact, the entire central nervous system is covered in these layers, the pia mater, this fluid, this arachnoid layer. The whole thing is like the entire central nervous system is in a bag with a little fluid on the outside and another bag. It's kind of double bagged. Fluid in the middle. And I'm going to go up here and just underline where it says the CSF. Oh, it's under the arachnoid layer around the exterior. And to take some blue, the CSF circulates around these spaces. So the light blue is the cerebrospinal fluid. I'll just kind of color code by underlining that. Now I'm going to take a dark blue and I'm going to underline the word veins. So drainage takes place either down through this area down here, all the way down into the spinal cord, all the way down to your tailbone, the whole way down your spine. In fact, they can put a needle in, right, and take some out, and they call it a spinal tap, and they can sample the fluid, You don't really want to be sticking a needle into the brain, but they can sample the fluid because this really fills up very quickly. I mean, this fluid will travel down and out in a matter of hours. So if they sample the fluid down here, they can see what's going on in the brain because that fluid was up here just a short time ago. So it can do that, or it can go into some veins that will drain into the jugular vein. So here's the vein in your neck, the big jugular vein. That's the main drainage vein. And the, let's see, how do we draw this? Under here, it can connect these to, there's basically it goes up and around. Now, we're only going to draw one. Let's see, because under here, like I said, this is not super highly accurate, but it gets the idea across. This is more in the dura mater. This is above the arachnoid layer, a little bit, right above it. Good. And then these would drain, maybe this one maybe it would be draining from this over here, draining. And there would be other things draining too, like this. But this drains into the jugular vein. Vein, plural. Don't want the S there, just jugular vein. And there's little knobs we saw in the one picture. Right here, the arachnoid layer has these little things that go out, and this is where the drainage occurs. The fluid gets pressed out of here into the veins, and like this drains like that. It doesn't show the jugular vein on this one, it just shows these. But so that's one drainage route. Number one is into this vein, and number two is down through that canal. Oh yeah, and they have names too. If you want to label this central canal, is called the central canal. And this, right here, between the third and fourth ventricle, this is called the cerebral aqueduct. You know, the aqueducts were those big stone water troughs the Romans made. So we got a big water trough draining from here to here. Reminded them of an aqueduct. And then let's add the skull around the outside like this, so that we know that all of this blue stuff is taking place between the cerebrum and the skull. And again, it's really out of scale, but and I'll leave you with this picture from lesson 51. It's good to refresh our memories about all the intricacies of the cells in the gray matter and the white matter. Of course, we didn't cover this in this lesson because we'd already covered it before, but don't forget that this is what is going on all through the brain, all these intricate little connections, and you've got the microglia and the astrocytes and all this. That's what's in all that gray and white matter.